Hello, today we are going to take a look at how we use variables in science experiments to make them fair. So first we need to take a look at a few basic definitions. Independent variable, dependent variable, and controlled variables. So independent variable is the one that the experimenter changes, the one that they're in control of, and I like to think of it as independent means input. So it's what the experimenter is inputting into the experiment. The dependent variable is the one that's responding to the independent variable. So depending on what it is that the experimenter is changing, whatever they end up measuring is the dependent variable. So it depends on the independent. Controlled variables are all of those different factors that are going to stay the same throughout your experiment to make sure that it's fair. So here's an example. Let's say a student wanted to determine whether feeder color has an effect. He might suggest, if I put up a red, blue, and green feeder, birds will visit the green feeder more as a hypothesis. He might speculate that the green feeder, being the most camouflaged or natural, might be visited the most. So in this case, remember that the independent variable is the one factor that is changed by the person doing the experiment. So here it would be the color of the feeders. That is what he is in control of. And then the dependent variable is the factor which is measured in the experiment, the one that depends on the independent variable. So that would be how much seed ends up being eaten out of the bird feeders. And then the controlled variables are all of the other factors that stay the same in the experiment, such as the location of the feeders, the kind of feeder that is used, and possibly putting the feeders out at the same time. So here's a little experimental setup for what this experiment might look like. And you can see that the independent variable are those three different colors, so that's the thing that's changing, and then the dependent variable, it's going to be the same. So in each case, we're going to always take a look at the amount of seed eaten. And you might also want to note then that that dependent variable is going to be a quantitative observation, right? It's going to be measurable. So if we have everything except the independent variable held constant, then we can say that our experiment is fair. So only the independent variable can be changing from one group to another to make it fair. So let's say that Sam wants to know if birds prefer one feeder of color to another. So he kind of has the same idea as the last one, but what he wants to do is make 3 feeders out of 2-liter bottles and paints one red, one blue, and one green. He fills the feeders with the same amount of sunflower seed and plans to keep the feeders out for 1 week before measuring how much seed is eaten out of each. On Tuesday, he puts each of the feeders in his backyard. The red feeder goes in a large dead tree, the blue feeder sits on the doghouse, and the green one he puts in a small bushy tree. So the question is that experiment fair? Well, remember, it's important to only change one thing at a time. And if we take a look at this experiment again, we see that not only is the color changing, but what else is changing? So hopefully you guessed that the other thing that's changing is the location of the feeder. So that would make this experiment not fair, or you wouldn't be able to trust the results. Here's another example. A girl wanted to know whether her dog being out in the backyard scared birds away. She thought it would because the dog likes to bark a lot and has been known to chase little animals. For 5 days when the dog was out, she counted for an hour. And for 5 days when the dog wasn't out, she counted for an hour. After she collected her data, she noticed that the birds are actually out more when the dog is in the yard. Does that surprise you? But remember, what the girl didn't think of was other things that might affect the number of birds that were out. She began thinking that maybe something else might be going on. She realized that the dog was usually out on sunny, warm days and not on rainy, cool days. So even though she found a change, how will she know if it was the dog that may have had the effect, or was it the weather? Here's another example. Patrick thinks that fish that eat microwave food are smarter than fish that do not. So Patrick times a fish going through a maze before and after eating. 10 of the fish get microwave food and 10 get non-microwave food. So here are our variables. Our independent variable then is whether or not the food is microwaved. The dependent variable is the time that it takes to get through the maze, again, quantitative observation. And then we want to make sure everything else is controlled. You want to use the same species of fish in each group, the same maze, the same amount and type of food. The only thing you want to change in between the two groups is whether the food gets microwaved or not. Now we're going to take a look at a Mythbusters experiment, and throughout the experiment you're going to be asked a few questions to try to identify some of the variables. So the myth that they're going to be experimenting, the myth that they're trying to bust, is the 5-second rule, that if you drop your food on the floor, as long as you pick it up within 5 seconds, it's good. There's not going to be any kind of contamination on it. And it'll feel a little bit like you're coming in the middle of their experiment, and that's because they've already done a little bit that you're not going to see. And they just tested some random floors in their lab, and they got a lot of different results. So they could have 2 spots that were right next to each other on the floor, and it would yield very different amounts of bacteria. So they decided that they were going to have to make their floor a controlled variable, so that you're going to see them set that up, and you'll get to see how they do that.
Do not try any of the experiments you are about to see at home.
You heard him. Don't do it.
In the myth of the 5-second rule, Adam and Jamie have decided to make a uniformly filthy piece of floor. That way, when they drop food on it, they should get consistent contamination.
So I see some pans,
We have some tiles.
final floor tiles.
We have beef broth. And we have some pans.
For some reason, whenever I see things, I have the desire to smash them on my head. You ready?
Yeah.
See if I can get a perfect face print.
So with that out of the way, what's the plan?
So the plan is that once we get these trimmed down to fit in pans, we're going to pour beef broth.
What's beef broth? Is it beef broth?
They leave the floor tiles to rot in the bibois, and Adam turns his attention to another mini myth.
I've always heard that a dog's mouth is supposedly cleaner than a human's mouth. I'm going to lick this sterile agar plate, and we're going to get Lulu the dog to come up and lick this one, and we'll just see whose mouth is cleaner.
So this has got nothing to do with the 5-second rule. We're just comparing your hygiene to a dog's, right?
Not my hygiene, the human's hygiene in their mouth to a dog's hygiene. I guess empirically what we will be testing is my hygiene against Linda's dog, but
Just do it. Just lick the darn thing.
all right, here we go. For science.
Ew.
Doesn't taste like anything. Where's the dog? Bring on the dog! Want to lick the sterile agar plate? Oh, Lou, that's perfect. Thank you. Good girl.
Is it just me, or does Lulu look quietly confident? Time will tell. Back at the myth of the 5-second rule, the floor tiles are fully festered.
What, 5 days now?
They smell really, really bad.
Yeah, I know. Let's get them out, hang them up, and dry them off, and see if we can use them today.
Jamie punches out plastic templates which will ensure sample sizes are consistent. While they're sterilizing in the microwave, Adam gloves up.
Coming up, Mythbusters takes Microbiology 101.
Adam has to behave like this. It's in his contract. But whatever you do, don't do this at home. To test the 5-second rule, they'll use a wet food, a dry food, and an evenly contaminated floor tile.
It's science.
Samples of wet pastrami and dry crackers are exposed for 2 seconds and 6 seconds. In other words, for less than and more than 5 seconds. They also do a control on samples that don't touch the floor at all, in case they are already coated with bacteria.
Control number 2. I know I love building big things and blowing stuff up, but this is also super, super fun.
Okay, fishing out a new one.
He doesn't care.
And go.
And the bulk of our Mythbusters bacteriology test is done.
Cool.
Can't wait to find out what happens.
So don't worry, in class tomorrow we'll take a look at the results of that little experiment. So that's it for variables in science experiments. What makes an experiment fair? We'll do a lot more practice with different scenarios and setting up an experiment using those independent, dependent, and controlled variables.